and I'm currently a research assistant professor in the Department of Microbiology and Molecular Genetics. Today, we're going to be doing lesson 1, an introduction to medical microbiology. So for the course objectives, we'll first start with the cultivation of lab samples. These are going to be the growth conditions you actually need to have in order to study these pathogens in the lab settings. We'll also talk about the equipment that is used to cultivate these bacteria. The second objective will be identification of the samples. These will be biochemical assays. This is going about how you actually would, in a lab setting, look and determine what kind of bacteria or pathogen a patient may have, and the tests that are available to get to these ends. Next, we'll also look at the evaluation of samples, and this will mainly be done by microscopy. So here we'll be looking at the visualization and recognition of key characteristics of each of these pathogens. So some basic equipment. The first one would be cleaning. We need to work in a sterile environment. We want to keep everyone healthy, and so for this, the main tool for this would be an autoclave. So autoclave, you can see, is shown here on the right. They do come in different sizes, so this is a personal lab autoclave. An autoclave basically uses heat, pressure, and steam to sterilize your environment. And so on the image, you can actually see you have a chamber that has a thick door to it. And so you would put your samples, this could be your tools that you're going to use in a lab, or if you actually need to kill something after you're done using it. You can put it in here, and then using, again, heat, pressure, and steam, you can then sterilize it. So an autoclave typically gets above at least 125 degrees Celsius, and this is sufficient for sterile conditions. A couple things to note about the autoclave, that it is not just dry air. And so when you look at the differences between trying to achieve a sterile environment with just dry heat versus actual steam, steam is considerably more efficient. So in about a couple of minutes with hot steam, you can achieve a sterile condition, whereas with hot air, it actually takes several hours. You should also note that if you're ever working around an autoclave, if it is finished and you're going to open it, you need to be very careful because very hot steam will come out. So you need to make sure it has depressurized and then very slowly open it before you get your samples out. Next, growing. So how do we actually grow the bacteria and pathogens we're looking at? There are two main ways. The first is for a fixed incubator. So here you can see this is just a 37 degrees Celsius chamber. On the shelves you can see petri dishes, which will contain the samples, which we'll talk more about a little bit later. You can also see the tubes that may hold liquid cultures. This door is also airtight and sealed, and then you can shut a metal door to keep dark conditions. The next type of incubator is just referred to as a shaker. So here you can actually see the metal rings, and in the back you can see a flask. So this is for when you actually want to take a growth media, which is a liquid nutrient broth. You can then inoculate new bacteria. You put it in here, and a shaker simply shakes and rotates the culture in a swirling fashion to help aerate it, to help the bacteria grow under optimal conditions. Next, we're looking at how do we actually see these things, and we'll be talking about microscopy. And the next lessons to come will actually walk through all the different parts of the microscope and how they can be beneficial to visualizing your bacteria. And finally, storing. In the lab setting, you may not always accomplish everything in the first day, and so the typical practice is to actually store things at 4 degrees Celsius. At 4 degrees, this actually slows the bacterial growth, which helps preserve your samples and keep them for long term. You typically just cram it full of many different things. So again, we see the test tubes that may have some nutrient broth and liquid culture. We also have petri dishes, which are typically wrapped in parafilm, which is just basically a waxy film that keeps it airtight. And you can also have different just test tube racks in the background. So general lab safety, a couple general lab rules that should always be followed. The first is that you should never eat or drink in the lab. This runs a severe contamination risk. A lot of the pathogens we're going to be working at are coming in through the hospital setting, so you never want to actually ingest that because you have open food in front of you. Next, we're actually going to look at personal protective equipment, how you can actually protect yourself while you're in the lab working on these things. So 3 main areas, the first of which will be what you will cover your hands while you're actually working. So here, you typically have 2 different options. First, you'll have latex gloves, which are the most common. However, latex allergies have been growing in recent trends. So we also, in most labs or hospitals, will offer a nitrile alternative. So most gloves that are either purple, blue, or green are probably nitrile. They obviously contain no latex and are very hyperallergenic. This is what you will wear when we do a vast majority of the lab practicums here. There are also 2 more additional gloves you can wear. You can actually wear them over a latex or nitrile glove. First, if for any reason you're working under extreme cold conditions, if you're working with liquid nitrogen or going to a minus 80 freezer, they have cold gloves. These are just basically long thermal gloves that you would wear in the winter. And finally, we also have thermal gloves for the other end of the spectrum for heat. So this is just a thick composite material. So this will be used for pulling things out of the autoclave to protect your hands, or any other if you're near a flame or if you have to handle any kind of hot device. Finally, we also have what we commonly know as the great white lab coat. This should be worn whenever you're working with chemicals that pose a threat to be spilled on. So you'll notice during the course of these lectures, sometimes I may be wearing a lab coat, sometimes I won't. If I am wearing it, that does signal that if you're in the lab, you should also have one on. But for the time being, we won't need it. One general rule about PPE, personal protective equipment, is that you never leave a lab with any of this on. You never want to walk into a public hallway, whether it's in the same building that your lab is in or not. All personal protective equipment should stay in the lab and be removed. So again, you should never be in a hallway. You should never go to the bathroom with your lab coat or any kind of gloves on, or especially to a cafeteria, anywhere else. You never want to see that. That's just a way of actually spreading whatever you're working on in the lab potentially to other outside areas. So for this course, you're required to keep a lab notebook. So I'm going to walk through just a general outline of how you may want to set up your notebook. It's highly suggested that you follow these guidelines. This will help you refer back to the lessons, the objectives that we have, and how to keep your results. So as you can see at the top title bar, the most common way to start your lab notebook is to give it an experiment number. And so typically that is done by starting with the first name initial, followed by your last name, and experiment number, followed by the title. As this is Portage Learning, for the duration of these lectures, I'll be abbreviating it using the initials PL. So as this is the first lesson, the experiment number is simply PL01, and this experimental section is keeping a lab notebook. And so before each lesson, you will start with an objective. And this is just a way that you want to write something down that's going to remind you what you were doing during this experiment. And so here, I've simply written to establish an organized template for keeping experimental records, procedures, and results. You can make this as exhaustive or as short as you want, but again, this is for your benefit. So the better description that you write down, the better off it'll be when you have to go back after a few weeks and know exactly what you did. Next, you want to have a procedure section. And so this is where you actually want to list the steps to any kind of procedure that we're going to be doing in the lab. So as indicated, you can see in step 1, this is where each step in the given protocol may be recorded. So this can be what kind of chemicals you use, what temperature, what growth conditions, what plates, anything like that. We're going to do this in a stepwise manner. One thing that is always recommended is in your procedural section, if there are any deviations that may occur from a standard protocol that may be given, you write that in red. It needs to be something that absolutely stands out. So if you don't get a result that you would expect, it may be due to a deviation in the protocol. And this should come obvious to you very quickly. You also have 2 additional sections. One is just a general section for notes. So any additional helpful information should be placed here. I may mention throughout the course of these lectures different things that may show up on the test or different notes that may not fall directly in the procedure protocol itself. And finally, we're going to have a results section. So as highlighted, you can see this is just where a brief summary of the final outcome of the experiment should be described here. This should be your go-to at the very end of every experiment. You should be able to go to it and know what is the main point of what you just did and what was the main outcome. So that's going to be the general overview of where we're going, and more lessons are to come.